the first experiment we're going to do is this Griffith experiment. And the Griffith experiment uses this type of bacteria called pneumococcus. And I don't know if you guys are aware of this, but bacteria of a given species, and species concepts are even weirder in bacteria, but bacteria of a given species can come in different variants, some of which cause disease and others of which don't. And you guys probably are already aware of this because you've heard of this bacteria called E. coli. Who's heard of E. coli? Okay. I saw people shaking their head. Really? That was some popular knowledge. So, do you have any E. coli in you? What do you guys think? How many think that you have E. coli in your body right now? How many of you think you have E. coli when you feel sick? Only about 3 people voted. Right? So that's not good. So E. coli is a resident of your gut, and it lives there and always lives there and always will live there. And you have many, many billions of cells of E. coli that live in you in harmony, at least, maybe with a peaceful agreement to not fight with your body. So E. coli is a regular resident of your gut. But when you eat the wrong kind of E. coli, and there's a few disease-causing strains of E. coli, they colonize your gut and make you really, really sick. And so there's a disease-causing variant and a non-disease-causing variant. When you go to Jack in the Box or you drink Odwalla juice, you get the disease-causing variant, and otherwise you just have the non-disease-causing variant. Pneumococcus, same deal. There's a type of it that causes disease, and there's a type of it that don't cause disease. The S type, which is growing on the right side here, this is just an image of a petri dish where these are growing little colonies. The S side is the smooth colony bacteria. It's just how they identify them when they grow in culture. And over here, the little colonies have kind of rough edges, and so they call them the R or rough bacteria. Same species, different variants. Disease-causing, non-disease causing. Okay? So for this bacteria, there's 2 variants, one that causes disease and one that doesn't. If you introduce the disease-causing one to a mouse, you guys are shaking your head, mouse torture up here. If you introduce the bacteria to a mouse and it's the S form of the bacteria, the mouse gets very, very sick and dies. And then you're able to culture that S bacteria out of that mouse. Question? Well, I don't know if it's horrible or not, but it is. There is a lot of experimentation that does use animals, and it's an unfortunate reality of a lot of medical science that whenever you want to scale something up to humans, you often have to figure it out first in a model system. And we're improving in our animal husbandry ways, so things aren't as bad as they used to be in terms of animal rights within the laboratory world. But in any case, if you take the S bacteria and you inject it, I've been practicing these mice for quite some time. On your little sheet here, those are not supposed to be bacteria. Those are actually supposed to be mice. Bacteria with heads, ears, and feet. So if you inject or introduce the S bacteria to the mouse, well, the mouse dies. An upside down mouse with an X in place of its eye is the universal acknowledged symbol for dead mouse. I think we all agree. If you take the R form of the bacteria and you introduce it to the mouse, well, it's not disease-causing, and the mouse is fine. Okay? Now, if you go and buy ground beef or something like that, and you got it at, I don't know, Walmart, you're a little concerned about its safety, who knows where it came from, what do you do to it to make it safe to eat? You cook it. And when you cook it, what happens? You denature all the proteins, you make it delicious, and it kills all the bacteria, right? So you take a meat thermometer when you're cooking meat and you measure the temperature, because once it gets up to a certain threshold, all of the bacteria die. So if you take these same S bacteria, and you heat them up until they are killed, so they're dead, it's probably no surprise to you that if you introduce those, I didn't give myself quite enough space here, to the mice, they're getting skinnier, smaller. There's no problem. The mice are fine. But this is where things get weird. And I think a lot of students find it to be suddenly intriguing and interesting. That is, if you take the heat-killed S bacteria and along with living R bacteria, you introduce them together, guess what happens?
Mouse dies.
The mouse dies. And not only does the mouse die, you can now recover from that dead mouse living S bacteria. Right? So if we take the S that's heat killed, we mix it together with the R bacteria. The mouse dies, and from that dead mouse, you can recover living S bacteria. So take a moment. Yes?
So we're introducing the R bacteria after the S bacteria has already been killed?
Yes, correct.
Okay, and this is in a petri dish?
This would be like cultures of living R bacteria and cultures of S bacteria that have been heated up till they die, introduced together into the mouse.
Okay.
Okay? So look at these data and discuss with your group what's going on here.